from ethical concerns, humans simply take too long to develop. Without animal models, we wouldn't even know that emmetropization exists. So here's an example. So here is a tree shrew, and you can have them wear these goggle frames. And we measure their refractions. This is days of visual experience. They're born with their eyes closed, so we start at day 0 when the eyes are open. Here's the refraction in diopters, hyperopic, myopic. Like children, tree shrews on average, this is tree shrews normal controls, they start out on average hyperopic, and then very rapidly over time, they converge on emmetropia from the hyperopic side. If at this point you take a tree shrew and you put a -5 diopter lens over one eye, with the lens in place, he's going to be hyperopic. Very rapidly, the vitreous chamber extends, the eye becomes emmetropic with the lens in place. Take the lens off, you've tricked him. Now he's a 5 diopter myope, but the axial extension of the eye restrains itself, and now the eye recovers and becomes emmetropic. The point is, the eye can detect not just the amount of blur, but its sign. Now, you can't determine the sign of defocus from an image, can you? You need to use trial and error to home in on sharp focus. The retina doesn't have that luxury. It needs to get it right the first time. How could it do that? My colleagues and I think that one of the main thing may be longitudinal chromatic aberration. So here, this is from human, this is the wavelength of light, here's the refractive error normalized to 589 nanometers, and basically you're relatively more myopic in short wavelength light and more hyperopic in longer wavelength light. It spans a little over 2 diopters across the visual field, across the range of visible wavelengths. So you can imagine here that if you have a myopic eye, the longer wavelengths will be in sharper focus than the shorter. That should be able to tell the eye, hey, you're too long, stop growing. On the other hand, a hyperopic eye, the shorter wavelengths will be in better focus than the long. That should tell the eye, hey, you're too short, grow more. So to investigate this, we use tree shrews, small diurnal dichromatic mammals that are very closely related to primates, and we alter the spectral properties of the ambient light. Here's just we use a Nidec autorefractor. We also use a Lenstar biometer. You can tell here we're measuring the left eye because you see the tail coming off on this side. And here we have tree shrews in narrowband red light. And what we do here is, this is again days of visual experience. Here's the refraction. Black is the average of shrews raised in normal white light. At this point, if you put them in the red light, wow, they go significantly hyperopic, and then they recover. Somewhat older animals, these guys have almost emmetropized. Wow, they go the wrong way. They become hyperopic and then recover in white. Remember, the red focus is farther back. If it was just following the sharpness of image focus, it should actually extend a little more. It's going the wrong way. Image focus without differential wavelength cues is not sufficient. This is not going to good focus. And a similar result's been found in non-human primates. I just want to point out here, this red light-induced hyperopia is due to a relatively shorter vitreous chamber depth. This is an axial hyperopia. We think that the red light is tricking the eye into thinking that longer wavelengths are in sharper focus than shorter wavelengths. Now, the two main things that can make animals myopic, wearing minus lenses and form deprivation, are largely eliminated with relatively brief interruptions. If an animal wears a minus lens or a form diffuser for 10 hours a day and you take it off for 2 hours, you basically cancel the effect and you have normal emmetropization. The red light effect seems to show a similar nonlinearity. So again, this is time. The black are the normals raised in colony white light. This is the tracing of animals raised 14 hours a day. 7 hours a day is basically the same. Here you got 4, 2, 1. Even 1 hour a day of red light and 13 hours a day of white light seems to produce over a diopter hyperopic shift. So could red light exposure be clinically valuable? Could we have kids wear red goggles, not all day, of course, but just for short periods of time? Red is a little dim, however. So when you put red filters on, things get really dark. So we thought, well, what about amber? If we have amber filters, say like this, they basically cut off everything below 530 nanometers. So it's much brighter, much nicer to look through. But it basically seems to have the same effect. Again, this is time, this is shrews and form deprivation. They go very rapidly myopic. If in amber light, nothing below 530 nanometers, a strong hyperopic shift. And again, this is form deprivation goes this way, the vitreous chamber goes much longer than the relative to the controls. With amber light, it's a significant relative shortening of the vitreous chamber. So could brief periods of wearing amber filter goggles have a significant clinical effect? Although some of the other data we have from tree shoes suggests that you couldn't just have tinted lenses, you'd actually have to block off the light around the side. A truly powerful control of emmetropization might require directly altering the chromatic image statistics as sensed by the short and longer wavelength cones. One thing we've hypothesized is multispectral multifocal lenses. So we're all familiar with the multifocal lenses. You have alternating relatively plus and plano zones. If you tinted these zones different colors, for example, if you tinted the plus zones blue and the relative plano zones more yellow, maybe you could magnetize and shift the chromatic aberration function. Maybe you could make something that is much more powerful at telling the eye to stop growing. So to sum up, myopia is primarily caused by our altered modern visual environment. I mean, we know there's a major genetic susceptibility component, but the main driver is our visual environment. If we had a better understanding of the visual cues used by emmetropization, surely we can change these cues or add new cues and stop the eye from growing too much. And while emmetropization, we think, likely uses multiple visual cues, our work suggests that chromatic cues are especially powerful. I'd like to thank my colleagues here, in particular Thomas Norton, who's worked on this for many decades. And really, this work is just based on what he has developed here. And of course, the Northern Tree Shoot, Tupaya Belengeri, and our funding, in particular, the NIH. And thank you for your time.
Hello, I'm Michelle Pardue. I would like to talk to you today about violet light and emmetropization. When we think about the visual spectrum, we're particularly interested in this very short wavelength, 400 nanometers, that seems to selectively stimulate a subtype of retinal ganglion cells that contain neuropsin or OPN5. And we have evidence that this violet light OPN5 pathway influences emmetropization and the development of myopia. I'd like to first acknowledge my collaborators, Professor Lang at Cincinnati Children's Hospital, and Professor Kurihara and Sabata at Keio University in Japan. Now, we know that outdoor activity has protective effects on myopia development, but the exact mechanisms behind this protective effect are not known. So one aspect could be whether or not the indoor lighting represents all the wavelengths that are found in sunlight. So here you see natural daylight, all the different wavelengths. But if I highlight just the very short end where the violet light is, you can see some of these bulbs, like the incandescent bulbs or the standard daylight white LED bulbs, do not have this short wavelength. Now we know that violet light is protective for myopia in chickens, humans, and mice. We also know that violet light regulates OPN5 and that OPN5 can photoentrain our local retinal circadian clocks in the mouse retina. And of course, circadian clocks have been implicated also in myopia. And we know that OPN5 mediates a light-dependent vascular development in the mouse eye through dopamine. And of course, dopamine is thought to be a stop signal in myopia. So we wanted to test the hypothesis that violet light OPN5 retinal circadian clock pathway is required for normal refractive development of the eye. So to do this, mice underwent lens-induced myopia and were tested under different lighting conditions. So at P21, the mice were fitted with this head-mounted goggle. One eye was exposed to a -30 diopter lens, and the other to a 0 diopter plano lens. The mice were followed for 3 weeks under different light conditions, and we evaluated them for refraction using a photorefractor or axial length and choroidal thickness using SD-OCT at P21 and at P42. So in the first set of experiments that I'm going to tell you about, we exposed mice to violet light at different times of the day. So here you can see the first control group. They were exposed to white light on a 12-12 light cycle, so 12 hours of darkness and 12 hours of light. The second group had white light plus violet light 3 hours prior to light onset. We call this pre-dawn. The next group had daytime violet light. During the light cycle, they also had violet light. We then have the continuous violet light group that had violet light under all hours of day or night. And then we have the white light plus evening violet light. So this is 3 hours of violet light right before the lights go off. And then we have 3 hours of violet light right after the lights go off. This is called post-dusk. All right, so let's see what the results are when we compare these different groups. I'm going to show you the myopic shift. This is the difference between the right and the left eye at the beginning of the experiments at 0 weeks post-goggling and at 3 weeks post-goggling. And you can see that our control group, our white light group, has a little over 20 diopters of myopia. Now, the other groups have myopia as well, but you can see that there's less myopia development, particularly in the groups that have violet light in the evening or in those dusk hours. And this is also mirrored in our axial length shift. Again, the difference between the two eyes, it is 0 when we start out, and then we have an elongation of axial length with white light. And you can see that when we have some exposure during those evening dusk hours, we get some protection. Now, the second experiment we wanted to do was to expose the mice to different wavelengths in these evening hours. And so we looked at violet light, blue light, green light, and red light when exposed between 1700 and 2000 hours. And when we do this, you can see that the myopic shift is only prevented with the violet light. And similarly, we only see a prevention of the axial elongation with violet light. Now, the next experiment was to see the contribution of OPN5. And we know that OPN5 is located in retinal ganglion cells. Here you see the axons, and here's a nice cross-section showing you the subtype. And these are individual OPN5 cells that have been isolated. So we used a retinal-specific conditional OPN5 knockout mouse with a CHEX10 pre-promoter, and then we mated that to OPN5 floxed mice. And the OPN5 floxed mice are going to serve as our control. So when we look at these mice, you can see that in the white light, or the white light plus the evening violet light, we have the shift with the white light, the prevention of the shift with the violet light. But in the OPN5 knockout mice, we do not see the protective effect of the violet light. And we see that both in the myopic shift as well as with the axial length. Now, we also wanted to see whether or not violet light regulated choroidal thickness. And first, I'm going to show you the white light, and you can see that here in the white light, we get a thinning of the choroidal thickness. And when we expose the animals to violet light in the evening, this is prevented. Now, when we do the same experiment with the OPN5 knockout mice, you can see that the animals still have a thinning of the choroid. So it was not prevented, this choroidal thinning, when the animals have OPN5 missing. So in summary, violet light inhibits lens-induced myopia in mice. The violet light exposure in the evening was most effective. And the protective effects of violet light appear to be mediated by OPN5. And in fact, this violet light OPN5 retinal pathway regulated choroidal thickness. Now, we don't know the exact mechanisms of this. It could be related to circadian rhythms or dopamine, but we are very excited about this violet light OPN5 pathway and feel that it could be a practical intervention to target myopia progression worldwide. I'd just like to end by again acknowledging my collaborators as well as our different funding sources. Thank you.
Hello, my name is Sally McFadden and I'm from the University of Newcastle in Australia. Today I want to talk to you about retinal pathways that contribute to the control of myopia in the mammalian eye. The translation of the signals from the rods and cones to the output on and off channels of the retina is modulated by the complexity within this inner plexiform layer and the amacrine cells. Today I'm going to talk about one particular type of amacrine cell that produces nitric oxide. In the retina, nitric oxide synthetase is an enzyme which catalyzes the reaction of converting L-arginine into nitric oxide, and in turn this changes the release of the inhibitory neurotransmitters in the retina. Like in other mammals, there are three different types of nNOS neurons. Type 2 project into the off layer, the displaced nNOS amacrine cells project into the on layer, and then the type 1 large cells here have most of their projections in the on-off layer in strata 3 of the inner plexiform layer. The distribution of these different nNOS amacrine cells varies. So the type 1 amacrine cells have a relatively regular array, although they decrease their density as you go from center to periphery, as shown here. But the displaced amacrine cells have an unusual distribution where most of the cells are located in the ventral retina. And that was confirmed too with PCR, where we looked at the expression of nNOS within the retina. Interestingly, this unusual distribution mirrors the distribution of the different cone types. Guinea pigs, like most mammals, are dichromats, and the highest density of these neurons mirrors the highest density of the blue cones in the retina. So today I want to show you that the density of these nNOS displaced amacrine cells is modulated by the degree, the sign, and the location of defocus. In order to manipulate the degree of defocus, we raised animals with Bangager foils, as published in this paper here, and these foils gradually decrease the high spatial frequency information that the eye can see. And as you do that, then the eye becomes longer and longer relative to the other eye, which is not wearing a foil. Of course, the eyes which are relatively longer are also relatively myopic. So this way we can change the degree of defocus and the degree of eye growth that we get in the eye. The increasing myopia that we observed as we decrease the spatial frequency information is not observed in animals which are raised with neutral density filters, which reduce the transmission of light to the eye, but do not interrupt the spatial frequency information. When we look at the difference in the nNOS displaced amacrine cell density, we see the same pattern of results, that we get the maximum reduction with the greatest amount of myopia, and as we lessen that amount of myopia, then that effect lessens. And again, we don't see this large reduction when we start to try and match the luminance only without the spatial frequency decrements. So these patterns of results are well correlated. So if you look at the correlation between the refractive error changes and the difference in the nNOS density changes, you see that they are significantly correlated. To investigate the sensitivity of these cells to the sign of defocus, we use the classic paradigm where animals wear a minus lens, which creates hyperopic defocus, so the image is behind the retina and the eye will elongate in order to compensate for that imposed defocus. Alternatively, if you make the eye myopic first and then remove that minus lens and replace it with a plano lens, then the eye will inhibit its growth as it tries to regain that compensatory mechanism. In control animals, we just raise them with plano lenses. So on the left here, we see the expected changes in response to wearing either minus lenses, which cause myopia and eye growth, or recovering eyes, which become relatively hyperopic and become relatively shorter in length. So what is interesting is that you look at if we look at the density of the nNOS cells, that we also see a sign-dependent response that correlates with this, and it's shown in more detail over here as a function of eccentricity from the optic nerve. So animals which are made myopic with minus lenses downregulate the expression of nNOS in these displaced amacrine cells. Planar lenses don't have any effect, whereas animals that are recovering then upregulate their nNOS levels. We also looked at the effect of whole field versus peripheral defocus on the expression of nNOS in these neurons. We found two effects of eye growth from the imposition of this defocus. First of all, most of the changes occur around the optic nerve and in central retina. That's the largest changes occur there, even when the defocus is across the whole retina. And secondly, the eye will integrate the defocus and average the defocus in order to drive what kind of ocular length difference the eye develops. When whole field defocus is imposed on the eye, then the nNOS displaced amacrine cells downregulate their density across the whole of the eye. However, when we put defocus only in the periphery part of the eye, these neurons only downregulate in the zone which is truly seeing that peripheral defocus. And in the central zone, they don't appear to be downregulated. Now, this is the area that is only being exposed to no defocus because that's the plano-only zone. 1% atropine eye drops inhibits myopia in our animal model, as it does in humans, of course. And what we find is that it also, if we give drops once a day for 10 days to young guinea pigs, we also find that it modulates the nNOS density. Specifically, animals that are given these atropine eye drops increase the density of their nNOS neurons, as is shown here. It is also the case that in the atropine-treated animals that the muscarinic receptor distribution is upregulated in the inner retina. It's also the case that these M2 receptors co-locate with type 1 nNOS amacrine cells. So here's a cell stain for type 1 nNOS, and here we see the muscarinic receptors, the same neuron. So they double label for both nNOS and M2. And if we look at the displaced amacrine cells, we can see that there is close apposition, as if there is an interaction between ganglion cells which have M2 receptors and these displaced nNOS cells. So in overview, we know that rapid light modulation activates nitric oxide release and changes the retina from one which is rod dominant to one which is more photopic and cone dominant, and enhances cone output. It also aids in light adaptation by changing the retina from a coupled state to an uncoupled state. This means that the retina, which has got widely coupled networks, is more able to be affected by low spatial frequencies and more blurred stimulus, whereas an uncoupled retina is more sensitive to detailed and local field information within the images. So a lot of this processing takes place here in the inner plexiform layer, and we've shown that these nNOS containing neurons which release nitric oxide into quite locally actually within the cells can then well placed to be able to affect the output ganglion cell on and off channels. And it is known that if you put in a nitric oxide substrate, for example, that you can block the off ganglion cell response. The effect of this wiring here is that ultimately it will sharpen the contrast that the cell's receptive fields, or the ganglion cell receptive fields, can see by increasing the surround inhibition. So whereas with low levels of nitric oxide, you see much reduced inhibition and a broader sort of field, wider field sort of stimulation output. So this kind of signal is a signal for the eye to become myopic and grow more, whereas this sharp vision signal is a signal for the eye to inhibit its myopia and in fact inhibit its eye growth. So in conclusion, I think that these nNOS displaced amacrine cells are sensitive to the local aspects of defocus, such as the degree sign and the location. And so they're like a local defocus detector. And it's also likely that they have high temporal resolution, given that nitric oxide is released in response to rapid fluctuations in light. Now, since defocus drives eye growth, these local detectors are obviously going to be an essential element in shaping the eye growth output signals from the retina. Finally, it gives me great pleasure to acknowledge the staff within my laboratory that have contributed to many of these studies. Particularly want to mention Hannah Bowery, who's now at Rutgers University, and also Grace Lee, who was a wonderful collaborator in some of the earlier studies that are reported today. And thank you for listening.
Hi, this is Dennis Chair from the Hong Kong Polytechnic University. The title of my talk today is the interactions between bright light and competing defocus during ametropization in chicks. This work was performed by Dr. Hui Jiang, a visiting student from Tianjin who spent 2 years studying in my lab. It is now well accepted that animal eyes are capable of undergoing homeostasis to regulate its size in response to the image plane. When a developing eye is exposed to induced myopic defocus or induced hyperopic defocus, the growth rate of the eye is decelerated or accelerated respectively. In other words, the growth of the eye is modulated to best match its focal plate. In clinical practice, there are already various kinds of myopia control devices utilizing the principle of myopic defocus. Those include ortho-K, specialty soft contact lens, and spectacle lenses. Generally speaking, they are up to about 50 to 60% effective on average. Apparently, the myopic defocus is not as effective in school children as in animals for some reason. Otherwise, they should have completely stopped or even reversed myopia progression. And that some important factors must be missing. On the other hand, there are substantial evidence that spending time outdoor protects children from becoming myopic. And there are a growing amount of evidence now suggests outdoor light intensities slow progression of myopia as well. So we came up with a simple research question. Can the anti-myopia effects of myopic defocus and bright light be combined to become even more effective? From previous animal study, we know that the bright light has an inhibitory effect, which is strong against form deprivation myopia, but mild against lens-induced myopia. For example, bright light of 15,000 lux slow down myopia growth induced by a 7 diopter lens in Regan Ashby's experiment. But somehow the treated group caught up with the control group in the later half of the experiment, so that there was no difference at the end. Similarly, the effect of high ambient light against lens-induced myopia was only mild in rhesus monkey. As you can see, the treated group and the control group show little difference at the end. So that's why we hypothesize that in those experiments, the effect of hyperopic defocus imposed may have dominated and mastered the effect of the bright light. And we propose that if we want to show any effect of bright light, we need to add some amount of myopic defocus. And so here are the two objectives of the study. To determine the effect of bright light on ametropization under mixed myopic defocus and hyperopic defocus conditions. And then to determine whether myopic defocus and bright light may be combined to enhance the inhibition of myopic eye growth. We selected 3 power combinations for the study. This schematic diagram shows how concentric dual power lenses works. In brief, the rings on the lens are of alternating powers, which altogether to provide 2 focal plates. In the case of a plano -10 diopter lens, the -10 diopter power produces a hyperopic defocus behind the eye. In the case of those plus and minus diopter dual power lenses, the 2 different powers produce a mixed defocus, 2 focal plate, that is. We use the metal halide lamp as the light source for the high ambient light, which produce a wide spectrum covering the short wavelength as well as violet and UVA. We apply the lenses on 5-day-old chicks and house them under a 12 to 12 light dark cycle. Control animals were kept under normal room lighting throughout the light cycles, while animals in the treatment group were exposed to 6 hours of bright white light in the middle of the light cycle. Lenses were applied on one eye, while the other eye was left untreated as a control. We measure refraction and A-scan every 2 days for a period of 6 days. Here is the result for the plano and -10 diopter group. You can see that the effect of the bright light produces a small amount but significant amount of hyperopic shift at the endpoint. Such difference was reflected in the reduced elongation of anterior chamber and vitreous chamber depth. No difference was found on the choroid. In the second lens group, the presence of bright light produced a stronger and more sustained hyperopic shift in the refraction, and the bright light treated animals showed relatively shorter anterior chamber and posterior chamber depth. In the third lens group, bright light abolished dramatically the myopic development and eliminated the relative elongation in the anterior chamber and the posterior chambers. By comparing the endpoints of different groups, we could analyze the effect of individual factors. First, when only hyperopic defocus was applied, the eye underwent a myopic shift of about 4 diopters. And we take that as a baseline. With the addition of a bright light, the endpoint was 2.8 diopter more hyperopic. With the addition of a myopic defocus, the endpoint was 7 diopter more hyperopic. If all factors were present, the endpoint was 8.8 diopters more hyperopic than the baseline. So we can conclude that the effects of the myopic defocus and bright light was additive, although not being synergistic. In a separate experiment, we compared the effects of different intensity of bright light. In brief, bright light of 20,000 or 2,000 or 40,000 K lux reverse the myopic growth to a similar extent, while bright light of 10,000 lux produce a partial inhibition of the myopic growth. In other words, there was a dosing effect, and that the maximum anti-myopia effect can be achieved by a light intensity somewhere between 10 to 20,000 lux for the species of chicken. So to summarize, bright light produced a relative hyperopic effect of the refraction endpoints in all treated groups after 6 days. The protective of bright light and myopic defocus were additive. The inhibitive effect of bright light was dose dependent and was attributable to and was manifest as some changes in both the vitreous and anterior depth, chamber depth, which is quite a unique features. So if a similar mechanism exists in human, there may be some potential clinical significance. Myopia management by supplementing myopic defocus or bright light alone may not be adequate to achieve full efficacy. And now data may suggest that children under defocus-based treatment for myopia control may be further benefited by exposing to bright light under outdoor environment. Those sayings should be tested by further clinical studies. This is the end of my talk. Thank you for your attention.
Hello everyone, I'm going to talk about the role of rod and cone pathways in modulating ocular growth and myopia susceptibility, and I'm going to show you evidence from animal models. There's no conflict of interest from any authors. So a large body of animal studies suggest that the normal eye growth is vision dependent, and the quality of image that forms on the retina really influences the nature of refractive development and ocular growth. Animal studies employ two common experimental paradigms. One is the form deprivation, where we put a diffuser in front of the eye and the eye develops myopia in response to it. And the other one is the spectacle lens defocus, where we put either a minus lens or a plus lens in front of a normal growing eye. And because of the plus and the minus lenses, it changes the position of the retinal image with respect to the retinal plane, and the eye develops compensatory ocular changes in response to the change in the image plane. So in normal growing eyes, when we impose a form deprivation or minus lens defocus, and when the light passes through the experimental manipulations, the retina is the first layer that senses the signals. It undergoes a series of biochemical cellular changes and sort of triggers a signaling cascade, which then goes through the choroid. It causes a thinning of the choroid, an increase in scleral growth that eventually leads to myopia. But the question we are asking today is that because retina is the first neurosensory layer, how are retinal pathways important in modulating ocular eye growth, and which are the important signaling pathways that might be important in that process? But in today's seminar, I'm just going to focus on photoreceptor pathways. The other important thing that to remember about the retina is that the retina itself synthesizes different neuromodulators like dopamine, retinoic acid, glucagon, and so on. And these neuromodulators can modulate the nature of the eye growth as has shown in different animal models. But today we are just going to focus on the rods and cones, and there is evidence from animal models in favor of both the rods and cones and how they might be modulating ocular growth. But these studies have been done in chickens, primates, and different mammalian models. But today we are going to focus on the mouse model and how the mouse model is important in our understanding of rod and cone dysfunction and their effect on refractive development and myopia susceptibility. The good thing about the mouse model is that because there are genetic mutations available, it ensures complete and selective blockage of the retinal signaling pathways we are interested in. There are hundreds of different mouse models available, but more importantly, when we impose a form deprivation or minus lens defocus in over the genetic mutant, it allows us testing for complex interactions between gene and visual environment. So I'm going to show you data from two different animal models, and in both animal models we employ two experimental conditions. One is the normal refractive development, where we measure the effect of the mutation alone without any visual environment manipulation, and we measure the refractive development from 12 to 14 weeks depending on different mutants. The second experimental condition is form deprivation, where we put a diffuser in front of the eye when the mice are 4 weeks of age and measure them at periodic intervals at different time points. And at each time point, we do weekly assessments of refraction, axial length, and different ocular parameters. We measure the refraction using photorefractor, and the corneal curvature using the keratometer developed by Frank Schaffel's team. And then we also look at axial length and different biometric parameters using biopterin spectral domain OCT. This is the form deprivation we do. There is a head pedestal, and from the head pedestal you can see that there is a goggle that is attached to the right eye. The important thing about this, or the good thing about the head pedestal, is that it can be easily removed so that it allows us to do those weekly measurements very quickly. So the first mouse model is the GNET1, and the mice have selective rod mutation because of the mutation in the rod transducin alpha, the protein that is important for rod phototransduction. We can see in this picture that the mouse have normal cone function, quite subnormal mixed rod and cone function, but completely absent rod function, suggesting that the mouse have significant functional deficits in the rod pathway, without any visible photoreceptor degeneration, at least at the young ages when the mice are measured in this study. This is the image of the refractive error development in the mouse model. The red line here is showing the GNET1, the dotted black lines are the wild type mice, and on the x-axis we have the age in which the mice were measured. It looks like the most prominent effect of the rod dysfunction was visible at young ages, at about 4 to 5 weeks old. These young mice, we can see that GNET1 dysfunction caused the refraction to be slightly more hyperopic compared to the wild type counterparts. But from about 6 weeks of age, the GNET1 and the wild type mice showed similar refractive development profile. On the right, we have the axial length. We can see that there was a significant increase in axial length with increase in age, but it wasn't really different between the two strains, which is quite common with different mouse models that we have tested. This image here is showing the effect of the form deprivation response. What I'm showing you here is the myopic shift, which is the difference in refraction between the form deprived right eye minus the contralateral left eye. The dotted line here are the naive animals that were not goggled. We can see that the goggled animals really developed about 3 diopters of myopia after 2 weeks of form deprivation, which was quite significant from the naive animals. But most importantly, the GNET1 mice, the form deprivation response was completely abolished, suggesting that the rod pathways are important in inducing the form deprivation response in these animals. When we looked at the axial length, there was an increase in axial length in the goggled eye, the opposite eye, as well as the naive animals, but these were not significantly different from one another. So in summary, the rod dysfunction disrupts the normal emmetropization and prevents the response to form deprivation, and rod dysfunction appears to be important for normal refractive eye growth in mammalian eyes. The second mouse model here is the GNAT2, and they have selective cone dysfunction because of mutation in guanine nucleotide binding protein that encodes cone transduction alpha, again a protein that is important for cone phototransduction. Again, we can see here that the light adapted cone response was completely absent in these animals compared to the dark adapted responses. In the picture on the right, we can see that there is no visible retinal degeneration at younger ages when we measured these mice. Here we are showing the refractive development profile. We can see that there was an increase in the hyperopic refractive error in both wild type and the knockout mice. The knockout mice are shown in purple, the wild type mice are shown in black. Then there was a further slight increase in the refraction for next 2 weeks, but the refractive development sort of plateaued for both strains from 8 weeks onwards. But most importantly, they were not statistically significantly different between the two strains. When we looked at the form deprivation response, the GNAT2 knockout mice appears to have a slightly greater form deprivation response compared to the wild type mice shown in the black dotted lines. There was a significant effect of strains suggesting that this response was slightly greater, but there wasn't any significant interaction effect. So in conclusion, the cone dysfunction does not alter the normal emmetropization under normal refractive conditions, and it causes a small increase in response to form deprivation, but there wasn't any significant interaction effect, suggesting that the cone function may not be essential for normal eye growth or response to form deprivation in mouse model. So in summary, it looks like the rod function has, or rod dysfunction has, moderate effect on normal refractive development, but a significantly large effect on form deprivation response, because we saw that the form deprivation response was completely abolished. But when we look at the GNAT2 mice, it actually had a small effect on both normal refractive development as well as the form deprivation myopia, suggesting that both rods and cone pathways might be important in regulating normal refractive development, but these two pathways have differential effect, and they both might be playing different role in regulating the eye growth and myopia susceptibility. Thank you very much for listening to me. I would like to acknowledge my collaborators and mentors, Professor Michel Pardieu, Mike Yvonne, and Richard Stone, and also my friends Hannah Park and Erica, who really helped me with data collection and putting these important papers out. I would also like to thank the APMS Congress for this opportunity to present my work at the Congress. Thank you very much.
I thank the organizer of the Asia Pacific Myopia Society for inviting to give a talk in this congress. And I'm happy to share with you some of the work that we have been doing on apolipoprotein A1 and eye growth in a cheek model. So I'm from Hong Kong. I'm CO2, Center for Eye and Vision Research, and also Laboratory Experimental Optometry, Center for Myopia Research, School of Optometry in Hong Kong Poly U. We know eye growth is a visually guided process, and it requires feedback from the external environment, and somehow the retina can receive these cues, visual cues, and decide which direction to grow. And in particular, in animal model, we know that optical defocus can drive different direction of eye growth. Myopic defocus can slow down eye growth, whereas hyperopic defocus can accelerate eye growth. And there are also corresponding changes in the choroidal thickness. So in myopic defocus, it will be thickening of the choroid, and in hyperopic defocus, it will be thinning of the choroid. And all that we know quite well in animal models. And in addition to the optical aspect of eye growth, and we are very interested to look at what's actually happening in the retina choroid sclera complex. What are the go and stop signals involved in the whole process? So of course, we know go signals is those or are those that will accelerate eye growth, whereas a stop signal will slow down eye growth. Well, in the published literature, there are a lot of those being proposed already. So the go signals such as PAC6, retinal exit, and hypoxia factor, and TGF-beta, and whereas a stop signal, you have dopamine and early growth response 1 and TGF-beta. And today I'm going to talk about apolipoprotein A1, which our lab has spent some time in looking at this expression. So far we know ApoE1 was downregulated in the LIM and FDM after a few days of observation. And you also know that increasing the ApoE1 expression using therapeutic means can inhibit or slow down the development of myopia in, again, cheek model. Also recently, we know that ApoE1 may bind to retinoid exit to affect its stop signals downstream physiological response. So when we look at ApoE1, we want to examine again how ApoE1 is being expressed in these models in terms of LIM and LH. Because LH we don't have a lot of published information related to LH expressions of ApoE1. So we have 3 groups of animals in LIH, plano, and LIM. So they were +10, plano, and -10 respectively. So we observed it for 4 days. So we record the usual ocular parameter using a scan and so on. And in terms of looking at the expressions of ApoA1 proteins, we use a Western blot and also a swab, which is a method of a mass spectrometry. So expectedly, the LIM and LH grows in terms of the change of VCD and axial length. So in the LIM, you have an accelerated VCD growth and also axial elongations. And whereas in the LIH, you have a retardation of growth, the ocular dimension, and also corresponding change in the refraction as well. So you have myopia in the LIM and also hyperopia in the LIH. So what about the ApoE1 protein expressions? And we found that there is a significant increase in ApoE1 expression in the LIH, but there is no change in LRN as compared to control. So you can see that, first of all, we look at the Western blot and there's significant increase in the LIH, and also we validate it with the mass spec data, and also we see the LIH and a significant increase in ApoE1 expressions, whereas the LIM, in both cases, in both methods, we don't see a significant change as compared to the control. So the next question we have is, can we manipulate the ApoE1 and look at its effects on normal eye growth? So we have manufactured this synthetic cheek ApoE1. So we inject those peptides into the eye from PN4 to PN6 daily, and we observe the changes in PN7. So it's a 4 days observation, and the cheeks are wearing plano lenses. So you can see that from the VCD and axial length and also the spherical equivalent, there is no change in the normal eye growth when we inject the ApoE1 as opposed to the sham injected control eye. So apparently ApoE1 does not affect the normal process of growth. But can it inhibit the myopic eye growth? So again, we inject the synthetic cheek ApoE1 peptides into the cheek eyes, so the intravitreal injections. So starting from PN9 daily to PN11, and then we observe changes to PN12. And the cheek is wearing the -10 and in both eyes. So the data show that ApoE1 significantly retard myopia development after 4 days. As you can see, the changes in VCD and axial length, and you can see there's a retardation of growth in VCD and also slowing axial elongations, and there's corresponding change in refractive error as well, as you can see that in here. So ApoE1 can slow down myopia development in cheeks. So in conclusion, we saw that the retinal ApoE1 expression was increased in the LIH, but not in LIM. And ApoE1 treatment did not influence the normal eye growth, but can effectively slow down eye growth in LIM model. So I think the data show that ApoE1 is indeed a stop signals to eye growth, and probably by manipulating ApoE1 and the related pathway might be another drug targets that one can explore further in terms of controlling myopia in the population. So finally, I want to acknowledge team members and also collaborators and also the funding sources. And I thank you for your attention.